Okay, so I'm going to talk about how we take these models and place them in our experimental data, which is either molecular replacement for crystallography or docking for cryo-EM. And our whole focus since about the time Phoenix started is using likelihood as a tool. So I'll tell you a little bit about likelihood and why we like that and how we use it for these processes. What's cool is, we've been working for ages on molecular replacement for crystallography. And then when cryo-EM became powerful and you started to be able to get high resolution cryo-EM maps, realized that actually a lot of what we learned when we were doing molecular replacement really applies very nicely to cryo-EM. Because you think of cryo-EM as giving you a real space image, but in fact both crystallography and cryo-EM work through Fourier space. When you're taking your particle images and combining them together to get a 3D image, you're doing that in Fourier space with structure factors. So a lot of the statistical analysis that we had been doing just transfers right over to cryo-EM, which is great. Of course, you have the phases, and that makes everything actually algorithmically and mathematically easier. So cryo-EM is a good thing to work on if you want quick results. So if you like thinking mathematically, they're both, when we do likelihood with cryo-EM or crystallography, both methods are based on understanding probability distributions of complex numbers. And if you are interested, there's all sorts of stuff. And if you're not interested, don't worry, because I'm not going to force you to learn that. So this is fairly simple. The basic idea of molecular replacement is, back when hemoglobin and myoglobin structures were done, it was obvious already that if you knew the structure of one protein, it told you a lot about the structure of a relative. And molecular replacement developed in the 60s and 70s as a way of saying, well, if we already know basically what our structure should look like, we can rotate it. If we could rotate it and translate it into the right place in the crystal, then we could calculate phases from that model and then put those together with the observed data. And that's exactly what we do. You take a model, and it turns out you can separate the rotation problem of putting it in the right orientation from the translation problem. So you take that model, you put it into the unit cell, and if it's in the right place, all of the symmetry-related copies get generated with the right relationships, and you can calculate phases. So that's easy. And people have always liked this when you could do it. I got involved in developing methods for molecular replacement as a PhD student a few years ago. And basically, I got involved in trying to improve molecular replacement because I was too lazy to do MIR. And I didn't like the idea of the nasty heavy atoms. Because I really like the idea of solving it from a single data set. So that's a big advantage. Now, the difficulty for all that time was mostly that you had, as Tom said, you had poor models, and now there's almost always a good model. So it's become much easier. And this talk is simpler than it was 3 years ago, because I don't have to tell you about how do you combine information from 5 different models at 20 to 25% sequence identity, because who cares? Right, but molecular replacement can still be difficult. And what makes it difficult is if you can only place a small fragment of what's in your crystal at a time. So if you have a very incomplete model, which usually isn't a problem with AlphaFold, usually you have pieces you can put together to give you a complete model. But if all the pieces are small or there's many copies, then getting started can be difficult. But it's surprising how small a piece can be, and you can place it. It's also, these are all different reasons where this would come up. And nucleic acid, of course, is an issue because, as Tom said, the nucleic acid models aren't as good yet. Of course, you also end up with poor data. So you have crystals that just don't diffract to high resolution, or they're very anisotropic, or you have various pathologies. So I'll say a little bit about these. Not so much about anisotropy, but you know what anisotropy is. It's when your crystal is better ordered in one direction than it is in another. And then your diffraction pattern, you get spots out to high resolution in the good direction and only to low resolution in the poor direction. So you get less information. There's also twinning, say a bit about that, and translational NCS, which is another complication. Our focus actually is on trying to make all of these things to handle all of these things automatically, so you don't have to worry about them. But it's helpful to understand them. And if you have a poor model, and as Tom said, there will be low confidence AlphaFold models, and sometimes the high confidence ones aren't quite right, but they're usually pretty good. But you do have to worry about whether you've got the wrong confirmation. Okay, I said we like using likelihood. And the reason for that is that you can take account of all of the sources of error and then you use the data properly. So if you know how accurate your data is, you know how well you should be able to reproduce it with a good model. And if you know something about how accurate your model is, you know how well you should be able to reproduce good data. And those both contribute errors. And if you understand those, then you know how much to pay attention to each part of the model and each part of the data. This makes it more sensitive than other methods that are of historical interest, like Patterson methods for molecular replacement. And it also makes it possible to exploit information from partial solutions. So if you have 15 domains you have to place, and you can successfully place one of them, that gives you a little more information for placing the next one, and so on, and you build it up. The other thing that in recent years we've found useful about likelihood is that the score that we try to evaluate is called the log likelihood gain. I don't remember if I have another slide, so I'll explain what I mean by that. The likelihood is a score which says what's the probability of measuring the data given my model. The better your model is, the higher probability the data would be. Kind of seems backwards, but it works really well. Now, the likelihood is a probability, and if you have probabilities of thousands of observations, you multiply them together, you get very small numbers. But if you take the logarithm of that, the logarithm goes up the same time the likelihood itself goes up. And the log of the likelihood actually has some other nice features mathematically. Then the gain part is you can say, well, I've got this number, but how do I put it on an absolute scale? And the gain is how much better can I predict the data with this model than if I said I know nothing. And knowing nothing corresponds to having a model with infinite errors. That's how you can approximate that. So log likelihood gain should be greater than 0, because 0 means you didn't learn anything. And the absolute value of that number actually means something. The bigger that number is, the more confident you can be that you have a non-random solution. And if the LLG, log likelihood gain, is more than 60, we found that usually means you're correct. And what's really nice is that same number applies in cryo-EM docking. In addition, we can estimate what the LLG should be from saying if our data, we know what confidence we have in our data because we have standard deviations of the intensities. And we have some idea of the confidence we have in our model. So if it's an AlphaFold model, you can say generally the parts that we keep for molecular replacement will be around 1 angstrom RMS error. So then you can estimate what LLG should you get, and then you can choose a strategy for solving the structure based on what you need to find an answer. And you can also choose between different possible solutions. Okay, so if we have a difficult molecular replacement problem, if you have an easy one, you just run the program and then you don't even have time for coffee, and then you've got a solution. So very interesting. If you have a difficult one, it really helps to have good data. Having higher resolution data really helps. And especially now with AlphaFold models can be compared to the highest resolution data. And you get more signal with good models. Don't forget about using anomalous differences. So if you collect your data set, if you have a chance of getting anomalous differences, like choosing a wavelength or collecting high redundancy data, go for it, because it's going to help you later. We'll show you that in the tutorial. And if you have pathologies, you have to identify them and figure out how to work around them. So if you have anisotropic data, you might not be able to do anything about it, but you could try different crystals or change the way that you grow the crystals. And the translational non-crystallographic symmetry, I'll say a little bit about later, but you have to account for that. And you use the ELLG to optimize the strategy. Right. Okay, so this I'll go through really quickly because Tom gave a really good explanation of all of this stuff about AlphaFold. So, I've always thought that it's just as important to know how confident you can be in your model and to have a good model. It's just as important. And the PLDDT score and the PE matrix make these models much more useful. Okay, that's all being covered. So we use these accuracy estimates, and Tom pointed out that the PLDDT, you can sort of relate that in some funny way to an RMSD. So there's a formula, we can translate it into an RMSD, and then we say, in our model, instead of saying this atom is here, say what's the RMSD of that atom, and if we just smear the atom out over all of those possible positions, then on average, it's going to be, the right position is going to have a reasonable density. So that's what we do. We smear out the atoms over the possible positions, translate the PLDDT into an RMSD, translate the RMSD into a B factor, and just stick in a B factor. And as Tom said, it's a real disaster if you don't do this, because all of your best parts of your model will have B factors of 90, and all of the worst parts will have B factors of 30, and the molecular replacement is going to be paying all of its attention to the worst parts of the model and ignoring the best parts. So real disaster if you don't do that. This is the example I was going to go through of something with lots of domains. This is the predicted model for fibronectin, which is got a lot of fibronectin repeats. And if you see the red stringy bits in between, the red stringy bits are probably disordered loops, but definitely we have no idea what they look like. So those we want to remove, but what about how well is the domain information known? Well, here's a PE matrix, and you can see that basically nothing's known other than those domains have pretty well-defined structure. And in the process predicted model, there's one technique that Tristan Kroll came up with for turning the PE matrix into domain parsing, and then it cuts it up into all those little domains for you. Yes, so I've talked about how likelihood is really sensitive for doing the molecular replacement, but that sensitivity is kind of a double-edged sword. If you have assumptions you've made in calculating the likelihood and they're wrong, it's also sensitive to that. So early on in the development of Phaser, we realized that it was really important to account for anisotropy if the data was anisotropic. So right from the beginning, we've been accounting for that, and that made a big difference. And we account for the possibility that the B factors of different components can be different. But then, yeah.
You're getting quieter and quieter as you speak.
Oh, sorry. I tend to do that. Okay. So we accounted for some of these things. And then what we found was all of the questions we were getting from people were, they'd send us data and we'd look at it and we'd say, oh, this has translational non-crystallographic symmetry. That's the problem. We couldn't deal with that. And it's found in, I don't know if it's more now, but at the time it was 8% of PDB entries. And there was a wonderful picture posted on the CSP4 bulletin board by Laurie Betts to explain what translational non-crystallographic symmetry is. So basically, it's when you have two things in your crystal which are not identical, but they're in similar conformations and related by a pure translation. So when I showed this picture in a talk, somebody then sent me pictures of his dogs, and they do that too. So apparently this is a common thing for dogs. Okay, so what happens when you have translational non-crystallographic symmetry? The problem is that, let's get back to our dogs. You have some diffraction from this dog, then there will be waves coming off of that dog that when it scatters the light. And if those waves are in phase with the ones that are scattered off of this dog, then they will be boosted up in amplitude. But if they're out of phase, they'll be canceled out. So what you see in translational NCS is your diffraction pattern does this. There's a very strong bit, and then there's a weak bit, and then a strong bit and a weak bit. And the main thing that's going on in your data set is that there's strong data and weak data. And that hides all the signal for the molecular replacement. So you have to account for that. And when we account for that, then we can solve really complicated things like this complex with 28 molecules in 4 sets of 7 that are related by translation. Twinning is when the diffraction pattern is superimposed on itself. Twinning we can sort of deal with reasonably well. It isn't as big a problem. Where it's a problem is when your data processing software and you get fooled by this extra symmetry that comes from the twinning and you put the data in the wrong space group. So sometimes if you have evidence of twinning, you should be looking for it. And you have to consider, do we have the wrong space group and do we have to try a different space group? So actually, this is what Airlie McCoy in the group has been working on for the last couple of years, and she's developing a new package called Phaser TNG. And if you're a Star Trek fan, you'll understand the TNG part. And it has modules called things like Picard and Scotty to do different things for you. And Picard will sort of automatically look at your data and figure out, does it have translational NCS? Does it have twinning? Could it be a different space group? And just go away and use all your processors and solve these things automatically. And that's getting really close to something we'll be able to give to you. Yeah, just a little bit about SAD phasing. So I don't know if you know about anomalous diffraction. Anyways, they're special atoms that interact differently with the X-rays at certain wavelengths, and they change the phase of diffraction. And this shows up as features in the data that you can use to deduce the positions of those atoms and then deduce something about your phases. So that's what's involved in SAD phasing. The theory of SAD phasing is actually very similar to molecular replacement, but instead of looking at one structure factor at a time, you're looking at pairs of structure factors. So we can use the same kind of theory and come up with a treatment for SAD phasing. And I'm not going to get into that, but the key thing is, because they're closely related, what we can do is we can do molecular replacement just looking at the normal data. And then we can say, where would we like to see anomalous scatterers to explain the differences between these pairs of reflections better? That's called a SAD log likelihood gradient map, and we can make that, and it will very sensitively show you where these anomalous scatterers are. So this is hardly, I mean, it's still used occasionally for solving structures, but it's more important now as a way of identifying what anomalous scatterers you have in your crystal. So if you have even chloride ions or sulfurs, but also calciums, or in the tutorial, we'll look at iron atoms. And then just a few words about docking and cryo-EM. So the docking problem is sort of like molecular replacement. We have data and we want to rotate and translate a model to explain our data. So how do we place an atomic model into the map? And we have to decide how are we going to score it. And of course, likelihood is going to be our answer to that. Most techniques out there are based on some kind of map correlations, but we find the likelihood to work better. And there's a search problem, and it turns out we can also do this with the same strategy as in molecular replacement, where you separately do the rotation search and then the translation search. And a key thing about likelihood is this decision problem people don't usually talk about. So if you do a correlation search, you get something that has the highest correlation between your model and the map. And how do you know whether it's right? With the likelihood score, it does turn out that if the number is bigger than 60, it's either right or it's wrong in a non-random way. So if you've got related chains in your structure, you might put the wrong model into a part of a map, but it's not wrong. It's just not as good a model as the right chain would be. Yeah, so we want to be able to deal with cases like this where you could just stick the map into Model Angelo and it would build a model, but it's nice if you have a model, be able to place it in there. This one at 7 angstrom resolution, you'd think it would be harder. It actually isn't that hard to place the domains in part of the map. What's really difficult is cases where you have something that's poorly ordered next to things that are well ordered in your map, and that's what, where the main challenges have been. How does the likelihood work in docking? Well, we need to understand where the signal and the noise are coming from. It's like the signal in molecular replacement is your model, and whatever the data tells you about that model, and the noise is the random errors in the measurements. In cryo-EM, the noise comes from the fact that you've got all these noisy individual particle images and you average them for many particles. The the signal can be reduced by lack of reproducibility in your sample because of radiation damage, or more importantly, often is having different conformations of your protein. And what we have to work with typically to work out what are the signal and the noise is the half maps. We can compare the half maps and say where they're similar, where they're correlated, that's telling us something about the signal, and where they're different is telling us something about the noise. And these are complicated things. So, we have, here's one of our favorite examples for docking because it's really a tough one. If you look at this particular PDB entry, this EMDB entry, they have plotted the orientation preference of the particle images that they got. And what you see is when you're looking from the side, which is like looking down towards this view of the molecule, there's lots of images there. So you're getting lots of information for the Fourier terms corresponding to what you see in that direction, but very few from other directions, which means your signal to noise ratio is different everywhere in the Fourier space. So we have to analyze that. And once we've done that, then we can do a rotation search, which is like a crystallographic rotation function, then a translation search, which is actually a single FFT, and use the expected log likelihood gain to choose a strategy, which I'm going to skip over. And we tested it, and it works really well. And we'll have an example of that in the tutorial. And the last thing I want to mention is, so we have the program that does the search over the whole map is called EM placement, or emplacement, however you like. But what we realized is that if you know where you want to look, instead of saying, I think this molecule is somewhere in my map, if you say, no, actually, I'm pretty sure this molecule is in this part of the map, you can do a much faster and more sensitive search. So this is in place local, and we'll see that being run in the Chimera X plugin. So it's really nice to run from Chimera X. And the YouTube tutorials Dorothy talked about, she's got a couple of tutorials to help you with that tool as well, how to install it and how to run it. And that's more or less it, yeah.